Greetings everyone. My name is Dharmendra Saraswat and I am representing Purdue University in the Symposium on Automation in Agriculture in Asia. I would like to congratulate the organizers for taking a bold step of moving the headquarters of Asian Association for Agricultural Engineering to India and launching a major international activity through the symposium. The topic is very timely, and I am happy to share my experiences with precision crop management by reflecting on a journey from single disease identification to multimodal sensing. My research efforts are primarily focused on four common corn diseases, namely northern leaf blight, gray leaf spot, northern leaf spot, and tar spot. The tar spot disease is of recent origin in the USA, which caused sort of an epidemic in 2018. As per estimates, this disease has resulted in lost production of nearly 500 million US dollar in Indiana in the last 4 years. To limit economical and yield loss, the very first step is to accurately identify disease before it starts spreading in the field. To accurately identify diseased plants during early onset of disease in the field, we evaluated a number of computer vision approaches, starting with image classification. Image classification helped us identify between the leaves affected by tar spot or gray leaf spot GLS or northern leaf blight NLB or northern leaf spot or NLS. A limitation of image classification is that it is unable to actually find out the location of disease plants in a field setting. Additionally, whenever there is a presence of multiple infected leaves within an image, those can also not be individually identified. So this limitation is overcome by another computer vision technique by the name object detection. So we performed one study with the northern leaf blight infected images where bounding box annotations were created for NLB disease, and then these were used to train the YOLO V4 object detection model that helped us locate and identify the presence of multiple NLB lesions present on individual corn leaves. Finally, we also experimented with semantic segmentation that helped us identifying the severity of disease infection on individual leaves. A review of approximately 70 research studies made it very clear that most of the research has been primarily confined to discriminating between healthy and diseased leaves or single disease identification on leaves. Whereas the reality is that multiple pathogens could be causing disease on an already infected leaf, and in that event, relying only on single disease identification could also result in incorrect disease diagnosis. Because of the tar spot epidemic in 2018, we conducted a study for diagnosing the destructive tar spot disease of corn. We relied on object detection and semantic segmentation deep learning approaches to identify disease lesions and also estimate disease severity on individual plants. The results, as you can see on the slides, were encouraging, and once they are incorporated with an automated sprayer, could result in treating affected plants more efficiently. As diseased or healthy crop discrimination or single disease identification present limitations when crops are infected by multiple pathogens, it is important to develop models for identifying multiple diseases. Therefore, we first acquired a custom dataset comprising images of multiple common corn diseases from the field settings. Then deep learning based image classification models were trained on this custom acquired dataset, and the highest testing accuracy of up to 99.41% was achieved for identifying multiple corn diseases. To accurately identify regions within individual leaves that correspond to different disease types, we resorted to slick segmentation approach that helps us split plant disease images into multiple superpixels, and each superpixel was then labeled as different disease types or a healthy pixel. And using this approach, we achieved accuracies of up to 97.77%. After the identification of multiple diseased regions, object detection deep learning algorithms were used to identify and locate disease lesions corresponding to multiple diseases. All this exercise was eventually combined with a two-stage approach where we first segmented the disease leaf and then extracted the diseased lesions from it. This helps us in estimating the severity by dividing the area of the lesions with the area of the leaf. Since the research in my lab is also focused on developing lightweight machines that could help in automating disease identification under field conditions, we also experimented with creating a novel neural network architecture that we have named as BNNDC, which is an abbreviation for Branched Neural Network for Disease Classification. And BNNDC comprises of branches within the neural network, and each branch is capable of performing a specific task. The branch on the first level distinguishes between different crops. The second level determines whether the crop is diseased or healthy. And finally, the different disease types are identified for multiple disease identification in the third stage. The BNNDC is a comparatively lightweight and achieved higher generalization performance compared to models reported in the literature. All the efforts that I discussed so far were based on close range handheld imagery, which is no doubt useful, but if our intent is to implement the deep learning models to analyze the entire crop field image, then those models need to be deployed on different platforms. And we experimented with a geodisease location system by using UAS imagery. And how we went about it is that in step 1, we first split the unmanned aircraft system acquired images into smaller tiles. Then in step 2, we passed those tiles to deep learning model that helped us identify and highlight disease regions in step 3. And finally, in step number 4, we created a progressive web application that had different screens where user could acquire actionable information that is coming directly from analyzing the US imagery data to implement some management practices on the field. A data pipeline was then developed to enable information retrieval from the unmanned aircraft system or drone, the unmanned ground vehicle or UGV, and handheld platforms so that a user is able to perform above and below the canopy corn disease diagnosis. We also developed a progressive web application and used a cloud database for publishing and pulling data from different nodes such as the handheld sensor, the aerial sensor, and the ground sensor. Overall, this multimodal approach has the potential to obtain disease status from the entire field to be used in a meaningful fashion for disease management. In summary, we have developed new data sets for multiple diseases, and this data set has been acquired under field conditions and publicly released to be used by other researchers or industry. We have also trained and evaluated several deep learning models for image classification, object detection, and segmentation with the aim to increase the accuracy of detection of multiple foliar diseases. We have also developed a new CNN architecture named BNN-DC that has improved generalization capabilities, and it has potential to be deployed on edge devices for automated disease identification. And finally, we have developed a data pipeline for exchanging data between the aerial sensor, that is a drone or a UAS, the ground sensor or a unmanned ground vehicle, or you can call it a robot, and also the handheld sensors that could be used by a user in the form of a progressive web application on a smartphone, so that an integrated and automated disease management system could be realized in the near future. Finally, I would like to acknowledge the research conducted by my graduate research assistants, namely Anis Ahmed and Varun Agrawal, my great collaborators, namely Dr. Ali Gamal, Dr. Guri Johal, and Dr. Brian Young, and the funding support received from agencies such as Foundation of Food and Agriculture Research, the Wabash Heartland Innovation Network, and the National Institute of Food and Agriculture Hatch Project. With this, thank you very much. If there is time remaining, I would be happy to answer any question. Once again, thanks to the organizers for giving me an opportunity to present, and I'm looking forward to working with the new association in advancing the profession of agriculture engineering in India and Asia. Thank you.